This is lab number 7, movement of molecules. Make sure you have that lab document in front of you so you can take notes during this video. So in this lab, we're going to be talking about different ways that molecules can move, and they move for lots of different reasons in the cells, and we'll talk about those some in here and some more in lecture. Okay. So in here we know that molecules can and will move across the cell or the plasma membrane. Okay. And they can move in a lot of different ways, different mechanisms of doing that. Some of those are passive transport, passive movement, which means that they don't require additional ATP to be added. Okay, they don't need additional energy. They can move and do that by themselves. Okay, and the other one would be active transport, which means it is going to require some addition of ATP. Okay, it needs some additional energy, some assistance to move where it needs to go. All right, so the first one we're going to talk about is a passive transport. It's called diffusion. It is movement of molecules from an area of high concentration to an area of low concentration. And the analogy that I give my class usually in lecture is that if you have something high up on a hill, okay, and you need to move it to the bottom, to the low part of that, okay, from high to low, it's not going to take a whole lot of additional energy. You probably just need to give it a little push to go, and then you are able to do that. Okay, it's not going to take a lot of input from you, really. No additional ATP or no additional energy. Okay, for that movement. Okay. The other one would be active transport, and we're not going to demo that in this lab. But the other one would be active transport, where you're going to be moving something from an area of low concentration to an area of high concentration, where there's a lot of it. Okay, or there's not a lot of it to where there is a lot, and so you're going against that gradient from low to high, right? And so if you're going to move that boulder from the bottom of the hill to the top of the hill, right, it's going to take a whole lot more input from you to get that moved over there. So you're going to need to require some additional energy, some additional ATP. Okay, so in here we're going to be talking about diffusion and passive movement. So the first one is we'll talk about diffusion, okay, movement from high to low concentration, and it's going to be affected by several different things, okay? How big is that concentration gradient? How big is that hill from high to low? Are you going to be moving that? Okay, that one's not in our lab, but the other ones that are in here are molecular weight, type of medium, what is it moving through, okay, or in, and then the last one is the size of the molecule. Okay, so molecular weight is kind of similar to the atomic mass of an element in the periodic table, right? So if you have an element, hydrogen, for example, in the periodic table, it is element number 1. It has a mass of 1, okay? And so its atomic number, or its atomic mass, and its molecular weight is 1, okay? This uses a standard, a chemistry standard called Avogadro's number. So in chemistry, they want to try to standardize how they're going to measure the units of measure. And so there's a scientist, and he came up with a standard unit, and that number is 6.02 times 10 to the negative 23. Okay? So why did I jump from that atomic mass? Well, we want to talk about molecular weight. Okay? Molecular weight is what is in our lab. And so we need to be able to understand that if an element, we'll get to molecules in just a second, but if an element, like hydrogen, okay, has an atomic mass of 1, it's also going to be 6.02 times 10 to the negative 23 Avogadros, right? Does that make sense? These units. So those would be the same, right? Hydrogen is 1, okay? So this is going to be used to define 1 mole, okay, 1 measure of unit of an element, okay? And so I'm not going to necessarily test you, like an actual test question on this number, okay? Maybe a bonus or something like that. But you don't have to memorize, per se, this number, okay? Unless you're taking chemistry, then it'd be really helpful. So go ahead and kind of plug that in, okay? So 1 mole, okay, of an element is going to be, okay, that. All right? And so when you start looking at molecules, let's say water, okay, it has 2 hydrogens and an oxygen, okay? What would be the molecular weight of water? Well, we would have to add those together. We would have to get the sum of those, okay? So I already said hydrogen is 1, so we have 1 plus 1 because there's 2 hydrogen, and then the molecular mass of oxygen is 16, okay? I can see that in a periodic table. I can go look that up, okay? So if I had 1 plus 1 is 2 plus 16 is 18. So I would have H2O, I would have the mass of 1 plus another 1 plus 16. That would be 18, okay? And that would be grams per mole, which would be why this standard number kind of came about for chemistry, to kind of centralize and kind of give one standard all around the world for those measures of units in chemistry, okay? So if you were to add all of the carbons, all of the oxygens, all of the hydrogens in a starch molecule, big, long, huge starch molecule, okay, it would be several hundreds, okay, several, or thousands, right, because it is a long molecule, okay, it's a big molecule. So how big, what the mass is of that molecule, okay, what its weight is, molecular weight, will affect it, okay. So I did an experiment. I started it before our lab, and so I took an agar plate, okay, and it has like that little gel, okay, bottom agar in there, and it's split down the middle, and I put 2 different types of stain, okay, of pigment, okay, on there. I have methyl blue on this side, and I have safranin on this side, okay? And so you can see that the size of the blot is significantly different. This is about 3 hours or more when we set this up in the lab, and then we just let it kind of soak into the gel, and then it kind of goes. And it's a little bit hard to see maybe in the lab, but right around the edge it's kind of shadowy, okay? So it is kind of spreading out, okay? This is diffusion, that's spreading from area of high concentration of the dye, right, of the pigment, okay, to where there's less. And so it's spreading out, okay? And you can see that this one is a lot smaller, okay, has not diffused very well compared to this one. And we did move it just a little bit as we were going, so it is a little bit out of shape, but it's still much larger. We didn't shake it around and spread it out like that. Okay, it's much larger, much faster diffusion rate for the safranin. Okay, and why is that? Well, if we look at the molecular weight, 438 versus 352. Okay, almost 100 grams per mole difference. Okay, it's a little bit less, right? But if I just ballparked it, right, almost 100 grams per mole less. Okay, smaller molecules, okay, or molecules with a smaller molecular weight are going to diffuse faster, okay? And I think that is actually one of the questions on our lab, okay? Diffusion is faster using a dye with a smaller molecular weight, okay, with a smaller molecular weight, okay? Methyl blue was 438 grams per mole, and safranin was 352 grams per mole. Okay, safranin is smaller. It's a smaller molecule. Okay, it has less molecular weight. Okay, so it is going to make a large blob compared to a small one, okay, as you saw in that plate, okay. It also depends on, diffusion rate also depends on what is it moving in, okay. So it can be moving in an agar plate. This is a little bit thicker than like Jell-O. I don't know, that sounds kind of weird, but if you've never seen an agar plate, you kind of need to know what that feels like. It's just kind of loosely rubbery, okay, a little bit like drying out Jell-O, okay, that kind of consistency. And that's what we're seeing in this one where we saw the dye, okay. Or we can look at that methyl blue moving in a beaker of water. Okay, and we can see how quickly, let me get my hand out of the way, we can see how quickly that might be moving in that water. And I'm trying not to shake it because I'm holding it. Right? Moving it around would make it go faster. But we can see it's already halfway diffused through this beaker of water in just a couple of seconds, a minute or so. Okay? So it moves very quickly. So in our example here, okay, and I'll pull that up in just a second to see when I see it all the way through. In our example here in our lab, diffusion is faster in a liquid medium than it is in a agar medium, okay? So the more dense, the more thick, okay, more viscous and solid-ish, right? That whatever it's moving through is, the slower it's going to move, okay? And so if I was to spray perfume, right, then that's going to even go faster. You obviously can't smell the perfume in the video, right, or the cologne, right, in the video, but it's going to move faster through the air than it would through even the liquid, okay? So if I was going to explain the concentration gradient that I said would affect it, if I do one spritz of cologne or perfume versus spill the whole bottle, right? There's a whole bottle of perfume or cologne up here, right? We're going to smell it much faster in this room, okay, much quicker, right, than if I just sprayed one, okay? And so you're going to have a larger concentration gradient is also going to help it spread, okay, diffuse much faster. And this is passive, right? This is all the way through now, okay? And so it only took a couple of minutes where this blue was like 3 hours or more, right? And so you can tell that it is a much quicker process, right? Depending on what it's moving in, okay? All right, so the next thing that we're going to do, this demo that I had explained the first two, okay? Molecular weight and the medium. And the next thing we're going to do is we're going to go through a simple diffusion where we're looking at the size of that molecule and whether that affects how that molecule can move or will move through a membrane. Okay, so I'm going to move this out of the way. All right, so in this one we're going to be looking at, let me erase this for us. All right, so in this one we're going to be looking at starch and iodine. Okay? So starch is a macromolecule. It's a polymer. It's many units of glucose bonded together, so it's a very large macromolecule. And then we're going to be looking at iodine, which is an element. Okay? It's a large element, but it's still an element. Okay? And so what we have, and I have a before and after. This is one of those experiments that has to sit for like 30 or 45 minutes, but we're not going to stand here and look at each other in the video. So I have already performed the experiment, and then we'll show the results in just a second. Okay? So what we did is we found, we got some, it's called dialysis tubing. It's just a plastic tubing, and it has very small microscopic pores or holes in it. Okay? And what we did is we put some water and some starch in one. I was looking for purple paper, but I don't think I have. All right, some paper. And we put some starch and some water in this one. And in the other one, we put some iodine and some water, okay? And then we tied off both ends to make this nice little package, okay? Let me fill them up, okay? The next thing that we did was we put 50 milliliters of water into 2 beakers, and in one of them we we put iodine, and in the other one we put starch suspension, okay? And we're going to look and see, this plastic dialysis bag is going to be our simulated membrane, and we're going to see, does the iodine move out of this one, or does the starch move in, okay? And then the reverse on here, does the starch move out and the iodine move in, or does both happen, okay? And we're going to find that out. And so what we're going to do is we're going to fill in part of our data table, and then we're going to put these into the beakers, and then we're going to let it sit for 30 to 45 minutes, and then we're going to come back and we're going to see our results for after the ending color, okay? So the first one we have is a starch bag, okay? And I already showed that. So we have starch and water in a bag, and starch is a suspension, and it is white in color, okay? And then the iodine is kind of an ambery color, okay? We looked at the starch reaction with iodine in lab, I think it was 4, okay, with the potato and that, okay? So iodine is an amber color, okay? Iodine and water. Whoops, I didn't talk about the beaker yet. Sorry. All right, and then the bag. The other bag has iodine, and it is amber color, okay? And so then we have 2 beakers. We have a amber color beaker, okay? And then we have a white color beaker, okay? And so with the starch bag, we want the iodine beaker, okay? So the iodine beaker is amber, okay? So we have starch and iodine in a reaction, and then we're gonna have iodine in the bag and starch in the beaker, okay? So we will put those together. So I have the iodine beaker and the starch bag, and I would put that in, okay? Just put it in, and then we're gonna let that sit for 30, 45 minutes. And then I have the starch beaker and the iodine bag, and I would just put that in, right? And it's gonna sit, right? And we're gonna allow diffusion to happen, right, for 30 to 45 minutes. And then we're gonna see what we get, okay? So I'm gonna set these aside because we don't wanna sit here for 30 minutes. Awkwardly looking, you know, in the video going what's happening, right? And so I have beakers where we've already completed, okay, the experiment, okay? So I'm going to look at the iodine beaker first, okay? So I had an amber color beaker because it had iodine in it, and I had a white starch bag. I had an iodine beaker, this is the one we just did, and a white starch bag, okay? That's what I started with. Now, the iodine beaker, still looking very similar, the iodine still looks very similar, so if I said it was amber before, it's amber still, right? Both of these look the same. Okay, but if I pulled out the starch bag, this was the before, and this is the after. Okay, this is the before, this is the after 30 to 45 minutes. Okay, and we see a dramatic color change in these. Okay, this bag now looks purplish. Okay, now it's purplish. Okay, so I'm going to put purple, right? It's a dark color, okay? So my explanation for this is, what does that mean? If you put starch and iodine together, starch is a color indicator for the presence of starch, okay? Iodine is a color indicator for the presence of starch, okay? So if starch and iodine come in contact with each other, we learned in lab 4 that that is a color change. It goes from amber to a darker color. I think we had black because we had a really high concentration of starch in lab 4. But it could go darker purple. And if I laid this down, if I have it next to this counter, it really looks kind of black. So I could say purplish black, okay? Purplish black, right? So that means that inside the bag, starch came into contact with iodine because I had a color change. So iodine did what? Iodine had to move into the bag from the beaker. That's the only place iodine was. It was in the beaker, right? It was in the liquid, right, that we have here. So it was outside of the cell of the dialysis tube, outside of the membrane, and then it moved in to cause a color change inside of that bag. Iodine moved into the dialysis bag. Iodine diffused, right? So I can say it moved in, I can say it diffused in. Okay? It diffused into the bag, right? And then caused a reaction. Did starch move out? No. Starch did not move out. How do I know that? Because I didn't get a color change on the outside. Iodine was there and iodine was there. If starch was present on the outside of that bag in the beaker, then I would have had a color change. But I didn't have a color change. So starch did not diffuse. Out. Did not diffuse out of the bag into the beaker. Okay? Otherwise, this liquid would not be yellowish amber color. It would also be purple. Right? It would also be purple or black. Okay? So we know that only iodine diffused right here. Okay? So the second one, right? So now we have our before, right? This is our before, starch on the outside and iodine on the inside. Okay? Starch on the in the beaker and iodine in the bag. This is our before. And then after 30 to 45 minutes, let me turn it around. After 30 to 45 minutes, now we have this kind of pastel-y purple, and if I spin it, some of it has settled to the bottom. We'll see that it will get a little bit darker. Okay? So we can see a lot of color difference there, okay? So the beaker had starch and water, okay? And it started out white, and it is now purple. Okay? What does that mean? I know I haven't answered that one, but what does that mean? That means that iodine had to move out. Let me go ahead and fill this, and then I'll answer both of those, right? So if we look at the bag, the dialysis bag that has starch in it, here's the before, right? And here is the after. A little bit different concentration, a little bit brighter yellow versus not as bright yellow, but not purple or black. Okay, so both of them still only have iodine inside of them. Okay, so this iodine bag started amber color and finished amber color. Okay, so it's kind of the same idea here, right? So it started out white, the starch was in the beaker, and then it came into contact with iodine and had a color change to purple. So iodine diffused out of the bag into the beaker, right? And changed that liquid to purple, okay? The iodine in the bag, some of it diffused out, so it can have a reaction outside in the beaker, right? But none of the starch moved in, and I know that because it didn't turn colors. It didn't change colors. It didn't turn purple or black, okay? So starch did not diffuse, okay? And we know that because those dialysis bags are the same color still. They're both still amber. Okay. Otherwise, we would have had a color reaction change. All right. So we have a couple of questions. questions to answer. And we kind of put it in our chart already, I think. All right, so we have it in our chart. Did starch or iodine move across the membrane bag? Right? Did it move across that? And I'm going to say iodine moved. And to be more specific, I'm going to say it diffused across, right? Because I didn't add sugar, I didn't give it ATP, I didn't hook up electricity, I didn't heat it up, right? I didn't give it anything extra. So it just moved from an area of high concentration to an area of low concentration. Okay? And how do we know that? Well, we know that because of the color indicators that we filled into our chart. Okay? Color change. Right? The color change that we saw here in our chart, where it only changed colors where the in one place. Right? In one place for each of those. And that's where the iodine would move where the starch was. Right? Iodine moving to area of starch. Right? But starch not moving where the iodine was. Right? So it's moving from highly concentrated area of iodine inside that bag to outside. Okay? Or high concentration outside in the beaker to inside. In the two different examples here. Okay? Why did one molecule diffuse and the other one did not? Okay? As you think about that, we scroll right here to the heading and the title of this procedure. Effective size of molecules on simple diffusion. Right? What are we testing here? We are testing the size of the molecule. Right? One molecule was smaller than the other. Right? Which molecule was smaller? Which one diffused? Iodine. Iodine is smaller. Iodine is the smaller of those two molecules. And the other one is larger, and so the only other one that we have is starch. Starch is a macromolecule. It's thousands and thousands of units of glucose bonded together to make this long string of a huge molecule of starch. Where iodine, although it is a large element, is just a single element. Right? And so it's a much smaller molecule than starch. Okay? So size of that molecule, right? If it's smaller, it will diffuse faster. Okay? The other idea some of my classes came up with was maybe starch was too big to fit through the pores in that bag. And we actually tested that and left it for like a week, and it still didn't move, right? So could be that it is too small, or it is the pores in the bag are too small. But regardless, right, we see a huge difference in that, where we have that diffusion of the smaller molecule happening faster, okay? Or occurring, right? So sometimes molecules may be too big to diffuse, okay, by passive transport. So maybe that starch molecule needed ATP. Maybe it needed to be active, okay? There's a lot of ifs that we can go through with that, okay? All right, the next procedure is osmosis, okay? And osmosis is a type of diffusion, okay? It's a little specific. It's diffusion of water, okay? It's diffusion of water, okay? And osmosis is diffusion of water across a membrane, okay? Across a membrane. I'm going to say that 1,000 times, right? To hint to you that when you're looking for a definition of osmosis, look for movement of water, or movement from high concentration to low concentration across a membrane. Across a membrane, across a membrane, across a membrane, okay? So osmosis is movement of water. And when we're looking at this, I actually want to move this down because I need some space. All right, so when we're looking at osmosis and movement of water, okay? Why would water want to move? Well, organisms like to be in homeostasis. They like to be balanced, right? They don't like it to be a concentration difference on some things, especially water, right? They want everything to be equally hydrated, okay, osmoregulated, okay? And so it wants to be balanced. So if a solute, if a molecule is too big to diffuse, let me do this one, too big to diffuse, right? Well, then something else may have to balance and move, right, to balance that pressure that's created, right? So if you're adding water to a water balloon and you just keep adding water, eventually that water balloon is going to burst, right? And so you're creating pressure in there. And so if that molecule, like starch here, was too big to move, right, there's pressure differences on those sides, and so you're going to need to balance that out. So if the solute, the solid particle, is too big to move across or can't move across, then the water is going to try to balance it out and equal it out, okay? Which way is water going to move? Is it going to move into or out of the cell? And that's going to depend on the tonicity of the solution outside of that cell, okay? And when I say solution outside of that cell, let me get one that you can see, okay? When I talk about the solution, the white liquid outside of this cell, if we said this was the cell, okay, this solution in the beaker, okay, what is the tonicity, right? What is that environment like, right? And so if I drew 3 beakers, and I know the definitions are here, but I'm going to define them by drawing a picture, okay? So if I could do 3 beakers, okay, and I'm going to describe and use these pictures to describe hypertonic, isotonic, and hypotonic, okay? I'm going to use these 3 pictures to do that, okay? These 3 beakers. So if I have a beaker, and I have a liquid inside of there, okay? I have liquid in each one of them, and I have a cell in there. Eukaryotic cell with a nucleus, membrane-bound nucleus, okay? So I have 3 beakers with a liquid, okay, and that's the one that we're going to be focusing on for hypertonic, hypotonic, and isotonic, okay? And I'm going to describe how osmosis works and how it can affect that cell in that environment, okay? So I'm going to start with isotonic, and I know it's not the order here, but I'm going to start with isotonic, okay? So I'm going to put isotonic here. The tonicity of the liquid, of the solution. So I'm looking at this liquid part, okay? The liquid, not inside of the cell. Inside of the cell, kind of an average human cell, right, there is a salt content of 0.9%, okay? So inside of the cell, if that's our cell, there's about 0.9%, almost 1% salt, okay, or solid particles, okay? So inside of the cell is 0.9% salt, okay? If it's isotonic on the outside, iso means same, so on the outside it's also going to be 0.9% salt, okay? So 9.9% salt. NaCl is table salt, is salt, okay? Sodium chloride, okay? So that means if it's 0.9% salt, then it's 99.1% water, okay? So in isotonic, it's going to be the same inside and outside. That doesn't mean water doesn't move, but it means that if one molecule of water moves in, then one is going to move out because it's balancing, right? It's already in balance, okay? It's already even, it's already at homeostasis, okay? Isotonic. This is where those cells like to be, right? This is their state of balance, right? For that, okay? If we're looking at hypotonic, hypotonic solution. Again, we're talking about the liquid part outside of this cell. The cell is in this solution. Okay, it's in the solution. The cell is still 0.9%. 0.9% salt, okay, which means it's 99.1% water. Okay. If it is hypotonic, hypo-low, that's my trick, hypo-low, it has a lower amount of salt, right, than the cell. Okay. Hypo-low. It has a lower amount of salt than what's inside the cell. So I'm going to say it has 0% salt, which means it's 100% water. Okay? So, if we're looking at this cell wants to be balanced. It wants to be the same. 0 and 0.9 doesn't sound like a whole lot, but it is to a cell at the molecular level. Okay? And so this is going to have to do something. So water is going to go through osmosis, and water is going to move from high concentration of water to low concentration of water. Okay? Hypotonic. Hypo talks about the amount of salt in the solution, or solutes in the solution out here. Okay? Out here in this liquid. Hypotonic has low solutes. Okay? 0. Okay? It has low solutes. Whereas the cell has 0.9. That's higher. Okay? So water is going to need to move in to dilute this 0.9 to where those are the same. So water would continue to move into that cell from high concentration of water to low concentration of water to try to get it to balance, to try to get it to be the same environment in both places. Okay? And so that cell is going to take on water. It's going to take on water, and that cell is going to continue to go, and then it will bust. Okay? This is a trick that one of my students came up with. Hypo will bust. Okay? If it's an animal cell, and that's the first one that we're going to see, okay? But it helps to remember even for the plant cell, and I'll show you that trick in a second. Okay? So it's going to, the cell is going to take in water and may burst, which is called lyse. Okay? It lyses, it breaks open. Okay? So that cell is going to swell up. Okay? It's going to swell up. Right? The opposite is true for hypertonic. Hypertonic solution. If someone is hyper, they have a lot of energy, right? Bouncing all over the place, they have a lot of energy. So hyper has high amounts of solutes. High solute concentration. Okay? So high solutes. So if the cell is 0.9% still, okay? It's the normal human cell, it's 0.9% salt or solute. Okay? 99.1% water. The solution has higher solutes. So it has higher than 0.9. I could put 1 and it would still be higher. I could put 5, 10, 50, right? 100. Okay, I'm going to put 10%. 10% salt on the outside, that's out here in this liquid. So if it was in here, this white liquid would have 10% salt, and inside of this bag would be 0.9. Okay? So higher amount of solutes on the outside in the solution, okay, outside of that cell, the outside environment. So if it had 90% salt, 10% salt, then it's going to have 90% water. Osmosis. Water is going to move from high concentration of water to a low concentration of water across a membrane. So water is going to leave the cell until or trying to balance these numbers. Okay? It's too concentrated out there, it needs to be diluted. Okay? It's too concentrated in there, it needs to be diluted. So it needs to add water. It needs to put water to the outside. Okay? So there's a lot of ways to word this. Hypertonic, hyper, high, right? Someone's hyper, lots of energy. High amount of solid, right? A salt in the solution that's outside of that cell. Okay? Versus hypo, low amount of solids or salts in the solution outside of the cell. Okay? And water's going to move to balance, okay, across that membrane. Okay? So I'm going to pause for a second so you can take a picture of that if you want, if you can still understand all of my scribble as we went through it. Okay? And then we're going to move on to, see that, I know you can rewind it and you can freeze frame it, but I'll give you a second. Okay? So in this situation, if water moves out of that cell is going to shrink, right, shrivel up, okay, and get smaller because it's losing its contents. It's like poking a hole in that water balloon and that water slowly runs out, or air in a balloon and it slowly will deflate. Okay, same thing here. Okay? All right. So here are our pictures, okay, that you have in your lab of showing a red blood cell, okay, or an animal cell. Okay? Animal cells only have a cell membrane, and so we're going to be able to see the shape of those cells, okay, or simulate the shape of those cells changing in our lab. Okay? And so this kind of depicts the same thing that I was just talking about. Okay? And so I'm going to move down to the next procedure. Alright, so I have 3 test tubes. Okay, and we're going to look at osmosis in an animal cell. And so we, the college orders factory from a lab, sheep's blood, okay? We don't go and get it ourselves, we order it from the lab. My students always ask me that, where'd you get that? Right, so we ordered it, comes in a bottle, okay? And then I have 3 solutions here, okay? Test tube 1 is going to have 0% salt, and what that is our distilled water, okay? Our distilled water, so it should only be water. There should be no salt, okay? It should be pure water, okay? If it's pure water, and it has 0% salt, okay, is that hypotonic, isotonic, or hypertonic? Okay, if it has low, can you get less than 0%? It's already 0. You can go negative numbers, but not when you talk about particles, right? So this is the lowest number. Low number, no percentage of salt is hypo, low. Okay, so this is hypotonic. Distilled water, okay, is hypotonic. Okay, 0.9% salt. I said that was the normal salt percentage in a human cell. That is isotonic. Okay, that's test tube number 2. They all look like clear liquids, right? Okay, from your end. We're going to see some differences in a second. And then 10%. This is exactly the example that I used when I drew those beakers, right? 10% salt is higher than 0.9, so it's higher than, so it's hypertonic. Okay? So what I'm going to do is I'm going to follow a procedure. I have those solutions in there. I'm going to take, and I actually added a few more than the 40 drops that it wanted, so it would be big enough for us to see in here. So I'm going to add like 6 drops instead of 4. I think it says 4. So I'm going to add like 6 drops of this sheep's blood to each tube. And I'm going to add them in order. That's fine. Okay. So 1, lab says, okay? And at first they might look all the same. Let me get them all set up and then we can talk about the differences, okay? 1, 2, 3, 4, 5, 6. And, oops. 6. Okay. Okay. So, I have 3 tubes. And when I look at them, right, probably maybe in the video, maybe it just looks red, right? Hopefully we see a little bit of differences in them, okay, from where I am at, okay? So I'm going to hold this up with some writing behind it, and we're going to zoom in. And so we want to be able to see, can we read that writing behind those, okay? Is it blurry? Is it see-through? Oops, let me go a little bit longer. Okay, so can we see through them, okay? And one of them we can, okay? This tube right here, we can see through that, okay? It looks like clear liquid. I don't want to say Kool-Aid because we obviously don't want to drink that, right? But it looks like a clear liquid or a see-through liquid with just red color, okay? Like a red, like Kool-Aid, okay? So we're going to see through that. This is test tube number 1. This is the tube that has distilled water in it, okay? If we look at the second one, it's a little bit opaque, okay? It's a little bit brighter red. Okay, and we are not able to read those words through that test tube. This is test tube number 2. This is in the 0.9% salt. Okay, so this is isotonic to the cell, to the red blood cells. So those red blood cells are still in there, okay, and they're kind of blocking our view and reflecting that color back at us, and that's why we're not able to see through it, okay? And then this last one, if I put these last 2 next to each other, they're both unable to see through. But the last one is a little bit darker red. It's the same, you guys saw, is same sample of red blood cells, okay? So why did we get a different color in that, okay? So what I want to do is I want to show you a little bit, and then we're going to talk about how to finish this table, okay? So what I'm going to do is I'm going to take 1 drop of each of these, and I'm going to put it onto a slide, and we're going to look at it in the microscope. One second. All right, so I'm just going to take 1 drop of each of these. Just give me a second to set that up. Get some cells in there. Just 1 drop. And the rest and go with the last one. Almost ready, hold on. All right, so I'm gonna put a cover slip on these, and we're gonna look at these cells underneath the microscope. Okay, and I'm going to try to find the test tube number 2 first, okay? So give me 1 second, I'm gonna find it, that way when we turn it on, it's all there for us. blood cells for this organism, which says sheep. Okay, so these are sheep red blood cells, right? And they're not flowing, and they're not sticking to anything, right? There's a few that are moving a little bit slower, but they're this nice round donut shape. Okay, I like to think of them, or I describe them as like a jelly donut, where the jelly has come out, and so they're not all the way through. It doesn't have a hole in it like a donut, but it's sunken in. So red blood cells, when they're mature, they lose their nucleus, and then that little indentation where it's a little bit lighter in the middle, right, is going to hold the hemoglobin, the heme group, where we carry oxygen, okay, so where the sheep is carrying them. So we can see these cells as nice circular donut-like cells, okay? Moving along, we can see they're slowing down, so we're running out of the liquid, they're getting warm underneath that microscope, okay? So this is the isotonic, okay? This is the 0.9% salt, okay? Solution, test tube number 2, right? And the cell inside is also 0.9% salt, okay? These cells are happy, right? In this solution, I won't say happy, but they are in homeostasis, they're balanced. It's the same salt content outside and inside of these cells. Isotonic solution, right, is 0.9% salt, okay? So these are the same. So what if I looked at, let me go to lower power so I can move, and we looked at the hypertonic solution. Back to high power. And look at those. I don't know how easy that is to see in the video. Okay. They are a little bit smaller, right? And if we search a little bit, this one maybe, here's one right here, right? Where they look a little starry shaped, right? Where their edges aren't quite as smooth and round. They're not quite that nice plump jelly donut style of shape. And if we continue to watch, they would get smaller and smaller and smaller. You can also see where it looks like some of them are sticking together, right? So once the size, once the shape of that cell, and probably the size too, once the shape of that cell, that membrane starts changing, right, they get a little bit sticky. They can stick to each other, they can stick to the blood vessels, okay, different things like that. And so they'll start clumping or coagulating and clotting together, okay? And so that's why when you look at this test tube, it's a little bit darker because we're seeing more light reflected back at us, okay? And they're clumping together a little bit more, okay? All right, so this is the hypertonic, didn't erase that. This is the hypertonic solution. And it was the test tube number 3, 10% salt. Okay. Where those cells are shrinking because there's more salt out here in the solution. Okay? So water is moving out of those cells to try to balance. Okay? So water's moving out, making those cells shrink, okay, and change their shape and get sticky. Okay? The last one is hypotonic. Go all the way over. And our slide's a little bit dirty. Go to high power. And try to focus. Alright, so I'm on it, but we're not really able to see anything. Alright, I have it on there. And it's on the slide, and if I continue to look around, maybe right there's some debris. Where did the cells go? Why, I didn't all of a sudden forget how to use the microscope, right? They're on the same one. So where did they go? Right? So if I have the hypotonic solution, which was the 0% salt, and the cell is still 0.9, those cells were there at some point, right? But water had to move into them to try to balance because it was more concentrated inside at 0.9 than it was outside at 0. Okay, so water had to move into those, and eventually that water balloon that we said, if you keep adding water to it, it will bust. Okay, so animal cells and their cell membrane can only withstand so much pressure until it's just too much, and then it busts. Okay, so those cells have all lysed, they've all broken open, right? And so when I said there might be some debris, some pieces of some cells, or maybe some that haven't broken yet, but I could continue to look and look and look, and we wouldn't find any, right? Like we saw in those healthy cells here, right? The healthy red blood cells, right? In the isotonic solution, okay? Where they're nice and round and a good solid circle where you can see this depression in the middle, okay? All right, so movement of water. All right, so back to our chart. This is a long one. Good thing you guys can pause, right? So we said this was isotonic. I'm going to abbreviate. We said this was hypotonic. And we said this was hypertonic. Okay? When we looked at the print, and I know we've already zoomed in a couple of times, we said test tube 1, I compared it to Kool-Aid. Again, we're not going to drink it, right? Okay? But we would be able to see that print through there. So, print visible? Yes. And I said the other ones were thicker, more viscous, opaque, right? And so no and no. You could not read through there, okay? So, I always look at the slide before I complete this chart because it's a really good visual of why can we read it and why can we not, okay? The last one I said we could look and look and look and we wouldn't see any cells. Well, there's no cells there to block our view to see through them, right? So this first one, where that water went into that cell and those animal cells burst, there's no cells there to block, okay? So the cells took on water, or took in water, I'm saying took in water, because they're in a hypotonic solution, until they burst. Okay, so they swelled up until they burst, and that process is called lysing. Okay. In these other two, the very first slide I showed you was the nice, pretty brown donut shaped cells, right? Cells are in homeostasis. They're in a environment that they're balanced. Okay, there's cells present, and those cells are going to reflect that light back at us, right? And then we see the red that they are sending back at us, right? And then the same thing here. Although those cells were a different shape, they were shrunk, right? Kind of pointy, right? But they were still there, and they were kind of clumping together, right? This one was a darker red. And so the cells are still there, cells are present, but they are shrinking. Okay, I think it's cremation, okay, is the word for shrinking, okay? Where they're losing water. Water is leaving that cell. Where here, water went into that cell until it burst. Okay? They're shrinking. All right, that is that exercise. Where's my cursor? Always technical difficulties. All right, the next ones we're going to see are going to be on a microscope, and we are going to look at this kind of similar process, but in a plant cell. Okay? And so plant cells going to have a little bit different structure on the outside of their cell than an animal cell is going to have. And so I have already pulled, we had a purple onion, a red onion, and I pulled the very thin layer, the purple pigment layer off of it, and I have a few of them on slide, okay, for us to look at. Right? And so we're going to prepare that, so I did that already, and then we're going to look at it underneath distilled water. So I'm going to try to gently put some water on here. I usually do that in front of my class, but I have a little dropper up here. So I'm just going to put 1 drop of distilled water on it, okay? And I'm going to then put a cover slip on it, and I'm going to look at this pigment, or this the pigment of the cell underneath the microscope. I put distilled water on here. What type of environment did I put this cell in? Distilled water is pure water, no solutes, so 0% salt. If you said hypo, then you are correct, okay? So hypotonic, okay? Distilled water is a hypotonic solution. That's the one that we'll use in here. All right, so I'm gonna put this on. See if I can find it. I got some air bubbles. I'm gonna try to find a clear spot where they are full. I'm gonna try to get close at first, and we may have to go back. That's too much. I'm trying to find a bright one. These all look okay, but I was trying to find the brightest one for you guys. They're on the slide. All right, I'm gonna go right here. See if I can focus on that one. Not coming in quite as clear as I want. Bear with me as I find a good one. This isn't something that you can plan. You just put it on there and then you find one. I'll try the other one. Maybe that one. All right, I'm going to use this one right here. It's not 100% clear, but I'm going to use this cell right here. Okay, so we can see that there's a clear edge. I'm going to draw it first and I'll take it off. Okay, kind of eyeballed it, right? So there's a cell right here. These are long brick-like cells, okay? And then there's some pigment color inside. It's kind of this lavendery purple, okay? And there's a little bit of debris on the slide, so just kind of ignore some of this black going around. But if we look at this cell, and we look at the pigment, and we look at the pigment inside of this cell, this pigment is stored inside the central vacuole, okay? So the purple is the central vacuole. And it's going to store this purple pigment, okay? That gives that red onion or that purple onion its color, okay? We see a kind of a white outline, okay? Plant cells have a cell membrane or a plasma membrane, and they also have a cell wall. Okay? They also have a cell wall. And so that cell wall is making that edge a little bit thicker, and then the cell membrane would be just inside of that. Okay? We have put this plant cell in distilled water, 0% salt. And you guys figured out that it was hypotonic solution. Okay? In the animal cell, in the red blood cell, it was the last microscope picture that I took. It was the first test tube where we could see through it, where the water went into that cell and they swelled up until they burst. Okay? In a plant cell, it's unlikely that they're going to burst, okay? Especially in our lab, okay? So we're going to see that purple, that central vacuole fill with water. Okay? Fill up with water, which is going to spread that pigment out all the way to where it is pushing on those edges of that cell wall. It might expand a little bit, and if we had maybe one more power, okay, a little bit more magnification, we may be able to see some of the plasmodesmata, some of the pores in between, okay? But it's going to fill up and push and put pressure against that cell wall, okay? It's going to push against it, okay? That pressure is called turgor pressure, okay? The pressure exerted on the cell wall from the inside, okay? If it is high turgor pressure, that means that it's filled up. It's filled up, and that central vacuole, that pigment, that water is pushing from the inside, just like it was on the animal cell, where it would swell up and stretch and stretch and stretch, except for that the animal cell burst. This one's not going to burst, it's just going to have more pressure, more pressure, more pressure. I guess eventually, like, there are some situations that it could, but it's going to be much harder to do, more unlikely to happen in a plant cell because of this cell wall. Okay? So this is going to be a turgid situation. Okay? This is a hypotonic solution, distilled water, on a plant cell. Water moves into the cell, expanding the central vacuole to push on that cell wall. Okay, filling it up, filling it up, filling it up, filling it up, push, 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 push. The cell wall says we're not going to break. Keep on pushing. Okay, and so that's going to create that pressure, that water pushing against that wall. That's pressure. Okay, so I'm going to take that same. I'm going to try a different one to see if we can get a better result. Okay, so the next one. Let's go back to this and answer our questions. Make sure I answered those because I think I did. View the purple onion with your microscope. Sketch a few of the cells on low power. Okay, that's what we just saw. And since it was a little bit, right, it might have some cells that look like that. Right, kind of brick-like stacked, right, and they're going to be completely filled in. That purple pigment is going to be all the way to the edge. Okay, it's going to be all the way to the edge for all of those. So you can fill those in, okay? So sketch a few on the low power. Are these cells plasmolyzed or turgid? They are turgid, okay? That water has moved in, and now it's pushing, it's filled up on the inside, and it's pushing against that, creating that turgor pressure, that high turgor pressure, okay? So let's go back, and we'll do the other one. And I need salt solution. And try to do a clean cover slip this time. Hard to tell. It's a long lab. Luckily you can pause and come back. All right, let's see if I can find some. Let's find some that are filled in. I have two samples in here. Let's try this one. They've been sitting a while, so they kind of faded. So this may take just a few minutes, but what I'm going to do is talk about, there's some air bubbles, those big circles are air bubbles. Because the sample is not completely flat. That onion will roll up a little bit. Just moved it to another spot. There we go. That's what we want. Much brighter. Let's see. In the middle. Okay. All right, so as this goes, it's going to take a few minutes to go. We have put this in a 10% salt solution. 10% salt. Inside of that cell is 0.9. So we have more salt on the outside. Is that hypotonic, hypertonic, or isotonic? 10% salt. Right? So this is 10% salt. This is hypertonic. I'm going to go inside. Right? So in the animal cell, in the red blood cell, that's when those cells shrunk. Okay? So if I were to keep watching some of these, right? In the last one I said it was completely all filled in. There was nice pink bricks, okay, solid right next to each other. Okay? We would over time see these shrink. And I kind of want to think I want to draw like right there. On the edge of that and maybe right there. See if while I'm talking, we can see the changes in those, okay? In those coming in. But we can also already tell that the space between these cells is wider than the other one. It's not working quite as quickly as it usually does. I always get those in my labs for some reason, okay? But what's happening here is this 10% salt is more concentrated outside of that cell. And so it is going to take water and it's going to move it out of the cell. Water is going to move out. And so that central vacuole is going to shrink, okay? And we're going to be able to see that in the pigment as it shrinks, okay? And I'm looking to see like where it's shrinking the best and see if I can find a better area. I can tell that it's not quite as dramatic as what I want to see, so I'm going to look around just a little bit more, see if I can find a better, see if I can, see how close I can get back to where I was. The one right in the middle is a little misshapen. Okay, and we can see, again, the space is much larger. And it's just one that's going to have to take a few minutes to go. Okay. All right. So we'll come back to this, let this sit for a few minutes, and then let's answer these questions, and we can go back and see if we see some dramatic changes. Okay, so we prepared the purple onion, okay, and we put it in 10% salt solution, and we placed it in a hypertonic solution. 10% salt is higher than the 0.9 that's inside of the cell. Okay. View the purple onion with your microscope. Sketch a few of them on low power. Hopefully when we go back, okay, we're going to still be seeing that cell wall of those onions. Cells. Okay, something like this. Okay, but we're going to see that central vacuole not pushing against the sides of that where that white space in between them is bigger than what we saw in that first one. Okay? So we're going to see that pigment not all the way filling up that entire cell, right? Where it is just parts of it, okay? Or where it's very small. Okay? And I'm just coloring in real quick. And this would be that solid purple pigment of that onion cell. Okay? So as that central vacuole pulls away from that cell wall, it lowers the turgor pressure. Okay? And when you lower the turgor pressure, it's called plasmolyzed. Okay? We're having them shrink away. And it's low pressure, right? The other one was filled up. The whole thing. That pigment was pushing against that cell wall. That's a lot of pressure, right? And now it's pulling away because water is leaving that cell because there's more salt outside of that cell than there is inside of that cell. Okay, let's see what we got. Give me just a second, let me see what I find one on the edge. Pigment left, I think I tried. And maybe a fresh onion. One more trick, hold on. That first sample of onion may have better pigment than the second. Sorry about the delay. Maybe I pull a fresh piece of onion. Well, the onion is not cooperating. Okay, let's see. I'll pull up a picture on the internet and see if I can do that. Always good times. It's just a second to pull up. Alright, so in the first one, right, the hypotonic, it worked pretty well for us. Right? We saw those pretty well. There was a little bit air bubbles for us on there. Okay, but we saw that pigment, that central vacuole, pushed all the way to the edge, filling up the entire cell. So this is a picture of a hypotonic solution environment for this red onion. Okay, and if I go to this one, maybe. it's not pushing against those edges. The edge comes all the way here for the cell, okay? Where here in this one, it is pushing all the way to the edge. Okay, here it's pulling away, and it could continue to shrink and shrink, okay, until that central vacuole becomes very small, okay, to try to balance out that amount of water, okay, for that. Sorry about the technical difficulties and the delay. Sometimes the microscopes in lab, sometimes they work and sometimes they don't. But luckily we got a picture here for us on the internet so we can see those dramatic differences, okay? Hypotonic, water goes into, and then hypertonic, where water is moving out of that cell. Turgid, plasmolized, okay? That is finally the end. So no, I'm sorry, we got 2 or 3 more things, so I've not quite the end. I think I closed it. Oh, 2 more questions. Phew. Really long. Okay. I was ready to be done too. Sorry. Okay. All right. So why did the red blood cells burst in the distilled water in the 0% salt, but the onion cells did not? Okay. What did the plant cells have that the red blood cells did not? They had a cell wall. That cell wall was holding that and withstanding and absorbing, taking that pressure, right? That turgor pressure, where the plasma membrane, the cell membrane, was just going to give, okay, and was going to break. Okay, when an amoeba, an animal-like protist, okay, burst in a hypotonic environment. Okay, so I put a picture of an amoeba, okay, from lab 6. We talked about the amoebas. In there, we talked about them having some specialty organelles, or you saw that they had some specialty organelles. But if this animal-like single-celled organism was in a hypotonic solution, what would happen? If it was in a hypertonic solution, what would happen? It's an animal-like cell. It does not have a cell wall. Okay? If it doesn't have a cell wall, in this environment, water is going to go into the cell, into the cell, and in this environment, water is going to go out of the cell. Which one would it burst in? Hypotonic solution, right? Hypotonic solution. It probably wouldn't happen as fast as the red blood cells that we saw because it has contractile vacuoles, which are going to help osmoregulate, right? But it still would burst, okay? That is finally now the end of this lab. It's a really long one. It actually lasts the whole class period as well. So thanks for watching.